Hi, I'm Allie Weber. I'm a postdoc at the University of Washington, and today I'm going to tell you about some of my work trying to understand the system that provides sensory feedback during insect flight. So all animals rely on sensory feedback to achieve controlled movement, and I think this system in the insect is particularly interesting because of how quickly this sensory feedback has to operate. So insect wing beats are typically on the order of 25 to 100 beats per second, or even faster than that. So the system has to be incredibly fast and precise in order to maintain stability. Sensory feedback from the wings is provided by a group of strain sensors called campaniform sensilla that are distributed over the surface of the wings. And so I'm interested in understanding what sort of information is being encoded by this population of sensory neurons. So the specific questions that we're interested in answering are: what sort of features are encoded by these neurons? Might there be different subtypes of these sensors that encode different features? And is it possible that individual sensors respond to multiple features of the stimulus? And that last question is really the focus of this poster. So to address this, we record from the wing nerve in the thorax using a series of multi-site extracellular recordings. So the wing nerve is the bundle of axons of the sensory neurons that innervate the wing. And then we also have a motor that we use to precisely control the movement of the wing, and a set of laser displacement sensors that we can use to track the motion of the wing with high precision and reconstruct the full surface. So we know exactly what was experienced at any particular location on the wing. So then we go through a standard process of spike sorting where we identify from those multi-site recordings the responses of what we believe are individual neurons, which we call units. And then from those neural responses, we can extract the features of the stimulus that are driving responses in those neurons. So we're trying to identify these temporal features, and all that a temporal feature means is this pattern or time history of wing. It can be either displacement or strain over time that is causing a neuron to spike. So we use a few different analysis techniques to identify these features. The most important thing to know about these analysis techniques are that these latter two, spike-triggered covariance analysis and maximally informative dimensions, allow us to identify potentially multiple features that might drive responses in the neurons, whereas reverse correlation, also called spike-triggered averaging, only allows you to identify a single feature. Also important to note is that maximally informative dimensions is the most flexible of these methods. It has the fewest constraints on the type of stimulus you need to use and the fewest constraints on what kind of features it can estimate. So using this set of analysis techniques, we identify a set of features that an individual neuron is sensitive to. So here I'm going to focus on the left-hand column, which is all this set of analyses for a particular unit, and the right-hand column is just the same for a different unit. So for each of these methods, we have a feature that or set of features that we identify from the method, as well as a function that characterizes how selective the neuron is for that particular feature. So when the stimulus is more similar to this particular feature, the firing rate is higher. The most important takeaway from all these panels is that using either spike-triggered covariance analysis or maximally informative dimensions, we find that there are multiple features that an individual neuron is selective for. So here you can see the two features that we find, and then here you can see that each one of these features does indeed modulate the firing rate of this neuron. And we see similar results for maximally informative dimensions. So interestingly, the features that we find with these two different analyses are fairly similar, but with maximally informative dimensions, we pull out somewhat higher frequency features that the neuron is sensitive to. And because this analysis method has fewer restrictions on the type of stimulus that you need to use and the types of features that you can find with it, it's likely that this is a more accurate reflection of what the neuron is actually responding to. So we've shown with a few examples here that individual mechanosensory units respond to multiple temporal features of wing movement. We can see that with either covariance analysis or maximally informative dimensions. So information-based techniques like maximally informative dimensions have not previously been used in this system, but they're particularly well suited to it given the fact that the structure of the wing tends to induce particularly complex stimulus statistics. And so these methods will be even more useful moving forward in the context of trying to understand how these sensory neurons operate in the context of natural behaviors. So thanks for stopping by. If you have any questions, I'm happy to answer them either via the Pathable app, or feel free to send me an email to the email listed at the top of the poster. Thanks.